Nice to have you here. It's a bit tricky to see you actually with the light. So just maybe also wave if you want to step in and have a question. I'm Timo Dickscheid from Jülich in Germany. This is Sebastian Bludau from our institute as well. So we do that together here today. And we will talk about the Brain Atlas and EBRAINS and how you can work with it with the different software interfaces. And also what the idea behind it is. This will be organized a bit as a mixture. At the beginning we will have a few slides just to get you into the concept, but then we will go into different hands-on examples where you will be able to follow what we are doing if you have a laptop and a recent web browser with you. So we will show you then links and you should be able to follow things in the web browser as we go along. We will also break at these places. If you don't want to do this, it's totally fine just to follow what we are doing. That should also be helpful enough. But if you have a laptop, just make sure you have a recent browser up and running, and then we will show you a few URLs as we go along. And please do ask questions as they come up. This is a tutorial, so if you cannot follow, if we are going too fast, just raise your hand and step in. We are not so many people, and the idea is that you can follow. So please let us know if that's difficult. Let me start by introducing very briefly this what we are doing in EBRAINS in terms of atlases. So EBRAINS is a research infrastructure that has been built as one result of the European Human Brain Project. And in the frame of EBRAINS, we have developed brain atlases of different species. There's, of course, an atlas of the human brain. There's an atlas of the rat brain. The Allen mouse brain is also included in the system. And we have developed an early version of a macaque atlas, which has been introduced very recently and is still growing and being extended. Here we will talk, this is the BigBrain Workshop, we will talk of course mostly about the human brain atlas and therein the big brain, which is a part of the human atlas. EBRAINS provides you access to all what we show through the web portal, which is at ebrains.eu. If you go there, you will see a page like this, and you can click on brain atlases and then get to this page where you find here on the left different categories. You find information about the atlases themselves, APIs, that's where you want to go if you want to do programming, integration to atlases, analysis tools. We will go into that. I just want to let you know that you find all of this also on the portal. Here we will guide you to these things. You don't need to go there now. So in terms of software tools, we have these three categories. Software for actually accessing the atlases, software for integrating data to the atlases, and there are also tools and workflows for analyzing data with atlases. This tutorial focuses on the one on the left, so on tools that allow you to access the atlases. The important thing that we have done in EBRAINS is, and this follows a lot of what Marianne Matone was saying in the very nice keynote this morning. We wanted to make sure that all the content in the atlases is actually well structured and follows the FAIR principles wherever possible. And that is why everything in EBRAINS atlases is, with a few exceptions, but mostly everything, is actually a data set with a DOI in EBRAINS, with rich metadata and coming in through the hands of trained curation persons, which are part of the EBRAINS curation services. So whenever we release a new map or anything, this will be a data set, it goes through curation, and it will have rich metadata attached, and this will be stored in the EBRAINS knowledge graph, which is a graph database following some predefined metadata schemes, in particular the OpenMinds metadata schemes that were also mentioned by Marianne this morning. The atlas services that we use to access EBRAINS atlases actually talk to these systems. You will not explicitly see this, it's happening in the background, but they actually rely on these systems and take the data from there. And you will typically always find, when you get a data element in the atlas, you will typically find this link back into the system to see the actual data set, underlying data set. And we consider this as very important. So we always want to make sure that that's the case. Of course, this infrastructure also uses machines, and so there is a network of high-performance computing facilities in Europe, high-performance computing centers that actually provide virtual machines, compute power, and storage systems to host all of that. And as always, you don't see that explicitly, but if that wouldn't be there, things wouldn't work. Anyway, the topic of this workshop here is challenges of big data integration, I think was the name. So it's important to mention that there is actually infrastructure needed under the hood to do all that. Let me now come to the Human Brain Atlas that we have built. The important idea of this atlas is that we really connect different modalities and especially different spatial scales. So we wanted to build a system that connects the millimeter scale, the surface world, as you have always seen it in depth in the NeuroMaps tutorial just an hour ago, that we connect these worlds defined mostly at the millimeter scale, whole brain distributions, macroscopy, down to the level of microscopy where you see individual cells and things like this. These are typically two worlds in terms of tools, in terms of data, in terms of ways that you work with them. And our ambition here was to connect these worlds and make that really easily tractable for users to work with an integrated system of both worlds. The way that looks like is that in an atlas we have regions defined. You see here the Jülich brain cytoarchitectonic atlas. This is here the MNI Collins space that we see here. For each of these regions, we typically have a map that encodes the variability of the region as observed in different individuals. So there is also already this dimension to cover the variability. And then we typically have examples for these brain regions mapped in a particular brain section, in particular postmodern brain at high resolution where you see individual cells. And we want to connect this into this system. And from there, you can typically, in a particular region of interest, zoom in and see individual cells, see cortical layers, study really the microstructure from there. So this is a path across the scales, not all the spatial scales we have, but quite a range already. The other level is, of course, different modalities. What I've shown here is mostly cytoarchitecture, cell bodies, and there are, of course, other modalities. So there are nerve fibers, genetics, connectivity, you name it. The idea is also to build similar connection to these other modalities. So we wanted to link structural functional connectivity from neuroimaging, from in vivo imaging. We want to connect histological measurements from different experiments to the same framework here to actually build such a interconnected framework, which we have actually also seen in the keynote this morning from a conceptual level. For this to work, we have to connect things. And there are different ways of connecting, for example, across the scales. And one way is, of course, to have different reference spaces, like a surface space, the MNI volumetric space. We also consider the BigBrain model as a reference space of a particular brain defined with very high resolution. And if we have brain regions mapped in these different spaces, this, of course, gives us a link, where we can select the same brain regions in the different spaces. Another more established link across these scales is, of course, to have spatial transformations between these different reference spaces, and a lot has been said and will be discussed also in this workshop in this regard. I think Lindsay is over there. She works a lot on these links. She knows that very well. Yeah? So these are two ways, corresponding coordinates, so to say, or corresponding anatomical concepts that could help us to make the connection. We have worked a lot on this, but the question is now how can you actually work with such a system and access it? And the approach that we have taken here is to provide an intuitive, explorative way of working with the system, as well as a way that allows to compute with this system. The intuitive, explorative way is, of course, to have viewers. So we have developed a web viewer that allows you to navigate across this system that you see there in a quite easy fashion, just using your web browser. The problem with this, and you can also download data there and find everything, the problem is that this is typically not reproducible, right? You do that intuitively, but it corresponds to your manual inputs, and you typically cannot nicely reproduce this, and it's also not easy to hand this over into a study and refer to this in your manuscript, things like this. So we built an additional way with a Python interface where you can do the exact same things, but in a purely programmatic fashion and highly reproducible and highly interoperable with other systems. We will talk exactly about these two things today and see practical examples. There are also analysis tools that directly make use of the workflow. We are not so much talking about this today. And there are tools that allow us to put in new data into the system, which is typically spatial anchoring. We will also not talk about this today in a lot of depth. So the two parts here on the top, the web viewer and the Python interface, they are actually on our side one software tool suite which uses a lot of shared code, and the one for online exploration visual is called Siibra Explorer. That's a JavaScript application mostly running in your web browser. The other one is Siibra Python, which is this programmatic framework. And they share a lot of code. So it's not two different softwares, it's actually two different interfaces to use the same software components. And that's it already mostly for the concept. So we will now go into the web viewer, see a few things, how you can experience this framework that I have conceptually presented in practice. We will then later on also see a few examples how you could do the same things in terms of programming with Siibra Python, and hopefully you will be able to see how this matches, you know, how these things correspond to each other. Just before we go into the online viewer, just to mention also that you understand how does data come in. There are different ways of registering data to an atlas. Two very typical examples are anchoring 2D sections into a 3D reference system, or anchoring 3D volumes of interest into a 3D reference system. And in EBRAINS we have built web-based workflows to do this if necessary in an interactive fashion. So for example, registering a tiny tissue block from a histological experiment into a reference brain is not something that as of today, as far as I know, you can reliably do in an automatic fashion. It requires many assumptions. You cannot do it 100% precise in different subjects, at least not in a human brain. So there are interactive tools to do these things. And I just want to make the point that I still consider this very important. You know, people who do image registration typically say, what is the value of manually putting a piece of tissue into a reference space that is defined from a different brain? But I think there's a lot of value. Just to show you a short example, we have here a piece of tissue that is basically just a visualization of a data set that's out there. And of course, if you have it like this, you would really have to read the documentation and make up your mind a lot to understand, okay, you know, which hemisphere is it? What is the size? What is close to this? And if you do a spatial anchoring into the BigBrain in this example, even if it's not totally precise, it helps a lot. Yeah, so we have here we would just load it into the system. That's of course not the correct position. And then you could here with this tool, although you deal with two large data sets, the BigBrain that you see underneath is in the terabyte range. This little volume is certainly in the gigabyte range. You can then interactively rotate, move it around, set the resolution until it actually sits well, you know, in an acceptable to an acceptable degree at the proper position. Once you have it there, it's much easier if you share it in the community, if you share this data set, if you have this connection, so people can see what is the size, what is the 3D orientation, where is it actually, what is close to this. That's an enrichment, and it helps a lot to better interpret this data set and also to discover it. All right, I think at this stage we move over to the to really see some real things happening. Sebastian will take this part and just show you the basics of the online viewer where you can discover this framework.
Yes. Hello, also from my side. Maybe just for the start. So I will go through the interface of the viewer, show you some of the important buttons. This is a Google document, and the address you can see written here in red. If somebody of you is trying to reproduce it, simply write it to your browser. This will bring you to this Google document, and all the important links we will need through this hands-on workshop can be found here in this Google document. So the link to the viewer, to the BigBrain viewer, everything is included here in this Google document. So if you want to follow yourself, please copy this link here. Okay. So let's go to the viewer. So yeah, this is, as Timo already introduced, the Siibra Explorer. It's the viewer for all the included atlases, which can be accessed through this viewer here. Most important, so here you have, it's volume-based, so we have 3 different representations, the frontal, sagittal, and horizontal cutting direction, and here a 3D representation. If you want to switch between different atlases, you can do it here by selecting this menu, and it's all built in the same way. So we have atlases. For all those atlases, we have different, not for all, depending of course on the atlas, but we have different templates which are comparable to the spaces, to the reference spaces, where you can work within the different atlases. And for all those different reference spaces, we have different parcellations. So this is the way the data sets are organized. So we have atlases, templates, and parcellations. As Timo already mentioned, we will here focus mainly on the human brain atlas. And what we can see here in the background is the Jülich brain atlas. This is an atlas which is based on cytoarchitecture. So we have analyzed 10 different postmortem brains for each of those maps, and created a probabilistic map for each individual area, which is displayed here. So let's select one here. This is, for example, area 4P. And at the beginning, we saw all maps displayed all over the template. This was a maximum probability map. In order to create sharp borders, we have calculated this one. But we also have individual maps for each areas. And this is one thing which we think is really important. So it's not only a label, it's really, there's a scientific study behind each of those maps which are included here in the data sets. And all the meta information, which we heard a lot of this day, are directly accessible through the browser here. So this is the representation of the map. So yellow means higher probability of being the specific area, and the more violet, the lower the probability is. Here you have a summary of the map which is currently displayed. You can also see it here in the breadcrumb navigation. You can navigate to the centroid of the specific maps which is selected, and you directly do have the DOI to the data set. And this is also one thing which is really important from our point of view, because all the data sets which are visible, downloadable, and you can find here in the viewer are really deeply curated. So you have all information you need to really work with the data in your own workflows. Description, again, of the data set, but also the references. Here you can directly link back to the viewer. You can download the data here, and with every data set you download, you get a data descriptor which describes the data and how to work with the data sets and where they're coming from. We have the individual publications. So there's one here for the concept of the atlas and the specific scientific publication for the area which is displayed at the moment. Okay, so going back here to the viewer. all those information you can have in a summarized version here on the left side. If you want to work with the viewer itself, it can be enlarged. You can zoom in, zoom out. So this depends on your needs. It's a typical viewer in this case. And but what's important here is that you can directly switch between different reference spaces. So we have started here in the MNI 152 reference space. If you prefer to work in the colon reference space, you can switch over to this specific reference space, and your position, so where you have been in the specific templates, is saved. So if you jump between different reference spaces, the Siibra Explorer tries to bring you to a position which is as close as possible to the position in the different reference space you have used. So is there anybody trying to work with the Explorer at the moment? So if so, maybe try to find language area, for example, area 44. So you can, there are different ways to access areas. So if you know where it's located, you can directly select it here using your mouse. You can also see the complete terminology, which gives you an access to all brain regions and how we organize them in the atlas. So you can select them here, or you can use this search option here. Not far too much. Okay, so now we have area 44 selected. So this is one of the ways you can work with it, but of course, yeah, as I said, you can also directly navigate through it and find it in the three-dimensional reference space. The same can be done also for different areas. So let's take, for example, area 4P, which is motor cortex area. And as you can see, it's loading. So it's here. And for this area, so since it's located down in the sulci, it's sometimes hard to directly find it with a pure volume representation. So therefore, you could also switch to the FS average, because here in this atlas viewer, both worlds are included. So normally, the Jülich brain atlas is coming from the volume world, because we also have a lot of subcortical structures included. But as we know, a lot of people are working with surfaces. So therefore, there's also a transformation of all those cortical areas to the FS average. And for example, if you are looking here for area 4P, it might be helpful to use the surface representation, and specifically the inflated version, if it's loaded. Okay, because here, as you all know, it's definitely easier to find because it's not hidden in the sulci if we use the inflated version. So from our point of view, both worlds, of course, do have specific needs. So if you really want to work on with cortical areas, sometimes access with surfaces is definitely easier. If you want to analyze subcortical structures as well, of course, you have to go to the volume. Here you can choose so which word you would like to use. Both are included. And so far, we have only seen the normal reference spaces, which do have a 1 millimeter representation. But what's also important, especially here for this workshop, is the higher resolution data sets which are included. So you can also directly switch to the BigBrain data set. So simply by selecting the template down here, and I've selected as a parcellation scheme the cortical layers, which were published by Konrad Wachsdühl. Also for those areas you find information. There's also, the reference is also hidden in the knowledge graph. So all the data sets are well annotated. And here we can see now the BigBrain model with its 20 micrometer resolution. But, oh, maybe I have to switch back because I've forgotten one important point here. The features. Wait a second.
Here you see how it preserves the viewpoint. So we are now zoomed in, and then we have an eye space. Of course, there's not much to see at this high resolution.
Okay, because so far I've only presented the maps and the meta information, but one of the very important concepts of this atlas here is the multimodal integration of different data sets. So therefore let's, for example, select here the primary visual cortex, which is a well featured area. And again, the probability map, all those descriptions, but there's a lot more. So you do have a whole bunch of features, regional features, which are connected to this data set. So those are, if available, they are really spatially anchored. Sometimes they're only semantically anchored. It depends a bit on the data set, but all those informations are written in the information files. So we do have a lot of cell density profiles. We do have laminar-wise cell densities and also subvolumes which are anchored in different areas. But there are also molecular data sets available, like the receptor densities. So for example, don't know, let's select one. Those are the receptor fingerprints or the receptor densities in general. Timo introduced on this slide for the area V1. And there's a basic visualization of the data sets, but of course you can also download them and work with the data if you want to use your own scripts or whatever. And all those features really depend on the areas, on the amount of information which is available. There's also a connectivity matrix included. So there's one data set from the 1000 brain study and also functional and structural connectivities from the Human Chromatome project, which are included. So yeah, you can really work on the structural, also on a functional level, depends on your need. But now let's go back to the BigBrain, because everything we have seen, as I've just said, was in a quite low resolution. But we do have a higher resolution, especially here in the BigBrain reference space. If we are now here in a region, so somewhere near the primary motor cortex, those layers we can see here, they were learned from machine learning algorithms and based on samples from isocortical patches. And this was done to produce cortex-wide surfaces of each individual lamina. And if you zoom in here with the 20 micrometer resolution, you can still see there are some cells in the background, but if you really want to go down to cellular level, 20 micrometer are not enough. And the same, like we have done here in the MNI space, there are different regional features. In this place here, it's spatial features, which can directly be searched through this browser here. So, for example, here in this is a normal cutting direction. Those are the reconstructed cutting directions from the BigBrain. And every yellow line here represents 1 micrometer scan, whole section scan, which was anchored in the BigBrain space. So it's roughly every 50th section which is included. And now we see those 11 ones which are here visible in the field of view. We have just zoomed in. Let's select one. So now I've selected section 3096. Oh, sorry. Okay, so it should not close it. So here, in this original cutting direction, we now really do have 1 micrometer resolution. And we still can see the laminas, which were automatically defined. And let's see if we can find something, because we said normally we should be somewhere around in the motor cortex. And if there's a motor cortex, I cannot find it at the moment. That's not the best section I've chosen here. So normally you should be able to see Betz giant cells, but let's try it again. So this should be possible to find them. Do you remember a section number which worked quite well?
Quite many, but I can give you one. Ist einer.
Here we have some, for example. So, typical for the motor cortex are those Betz giant cells, which are normally located in the lower lamina 5 of the isocortex. And here, if you superimpose those laminas, you can see that they are directly located at the border between lamina 5, lamina 6. And this is the place where we would suspect to find them. Here it's completely in lamina 5. So there's a real benefit of high resolution because you can zoom in down to individual cells. So this is the maximum resolution here. It's 1 micrometer in plane resolution, and those Betz giant cells are the ones we are looking for. But as we have heard during the introduction of the concept, so the viewer is one starting point to go into the data sets. But if you really want to work with the data and analyze it, of course, you would like to analyze it in a programmatic way. So simply viewing the data is nothing which works for a publication. And therefore, you could, for example, if you want to annotate, or if you have selected, if you have found a specific region of interest, you can annotate. Select the annotating tool here on the right side from the menu. Here you can select a polygon and then select points. Unfortunately, you cannot see what point you have selected, but so you have to believe me that there's a polygon behind. And so we have selected a region of interest here. And if you want to work with these polygons you have selected here in a programmatic way, you can export your annotations, which at the end it's a JSON file giving you the coordinates and reference space and everything you need to analyze it in a programmatic way, which then can be done in the Python world, which will again be presented by Timo. Oh, have I forgotten something? So then please add it.
Yeah, I take over from here. So just to say this, we recently shipped an update, and in this update, the points where you annotate are actually below the segmentation mask. I think this will be fixed by tomorrow. It is already raised to the developers. But you can really click here, and then you get here a representation of the annotation in a JSON format, which is actually for those who make sense, already uses this OpenMinds metadata scheme that I also said is underlying the EBRAINS knowledge graph. I think these polygons here do not, but you get a JSON representation that you can store to disk or copy over. We will do this now to show you how you can programmatically work with such things. Because when we first designed these tools, we found it quite tricky. Yeah? So here, this was an interactive way of going down from the macroscopy to the cell, and this was mostly zooming in. But now, how are you doing that when you're programming? It's not straightforward. And the basic idea in the programming workflow is typically that you start with some form of location information. That could be such an annotation. It could be just by selecting a brain region by its name. It could be by having an image with a peak in it, let's say an fMRI activation map. All of these things give you some form of location information, right? There could be a peak, there could be a bounding box. In the end, there is some form of location information. And of course, this information you can then use to go across the scales. You are then facing the problem that all the data structures that you are dealing with have to be safely connected to the reference space where they are coming from. This was also touched in the presentation of neuromaps in the morning. So you have this problem that, well, if you have a volumetric image, for example, you need to take care in which space is it defined. If you have a point coordinate, you need to take care in which space it is defined. Actually, you should also take care what the precision of that point is. It is very different whether you have a point that, for example, represents the contact point of an electrode in the brain, which certainly has an uncertainty of 2 millimeters or something. I'm not an expert there. Or whether that was the centroid of a segmented cell, which is defined in the micrometer range. So it is important that all these data structures that represent a location in the brain have properly attached the reference space that they are from and also the resolution. And we try to take great care that this is implemented in the programming framework that we use here. So let's have a quick look at that. This is not my computer, it's Sebastian's. So I will have, do you have it opened already? Well, let me just show it. So first of all, we have some tutorials on GitHub, and the link for those is on the Google Doc that we showed in the beginning. But if you, I think you can also just Google for them. It's a GitHub repo called Siibra tutorials. I think if you just Google Siibra tutorials, you also get there. And here are a range of tutorials, which we are currently just migrating to the new version. So as of today, some of them might not work properly. We have just released a new version of Siibra. But there's also one here for the BigBrain Workshop. And if you click on it, you can already see the notebook. But you can as well just open them as a notebook running in the cloud. So here below is a table, and if we look here at the number 8, that's again the BigBrain Workshop one, and you can launch this here directly in MyBinder, which is a cloud service for running notebooks, or on EBRAINS. This morning MyBinder was down. That's not maintained by us, but it didn't work for me this morning. They had a problem, it seemed. You can also open in EBRAINS, which requires you to have an EBRAINS account. You can get that in very quickly by just going to ebrains.eu/register if you don't have one. If you want to follow this, you can also try MyBinder. Usually it works. I go here to eBrains, select a supercomputing center. Or did you have it open already, Sebastian? Because that usually takes, well, here we are. So we have the notebook opened here. Is everyone here at least basically familiar with Python? Yeah, so, okay. So let me just guide you briefly through this. This is a short notebook. It doesn't show everything you have seen in the viewer. It just shows a specific thing, namely, how do I actually get a piece of image data at full resolution with the individual cells in the BigBrain. So what we are doing here is we, as usual in Python, we just we update and we import the packages. So that's here Siibra, and everything else is just standard packages that you might know, matplotlib, nylearn for some plotting below. And what I have done here is I have just copied over the string representation of the JSON file with this annotation we just did in the online viewer, right? So here, that's not the exact same annotation we just did in the viewer, but you could simply paste it in here as a multi-line string into a variable. I call it variable structure, it doesn't matter. And you see here that Siibra can directly create an object from that structure. Yeah? So I can put that JSON file in, say Siibra.fromJSON, and it will recognize this format, and it will know this is a set of points from the annotation. So when running this, you should get a point set. And one thing that you see here already, if I just print this point set, you see it knows already that this point set is defined in the BigBrain space, since that's part of the JSON specification, and the viewer has already tracked this. So we know this is defined in the histological space of the BigBrain model. And here in my annotation, I think it's four 3D points that make up the annotation. And now I have them as an object here available in Siibra. And in order to access image information, what I'm doing here is I just say my volume of interest, you know, we are working here in 3D, my volume of interest is the bounding box of these points. Yeah, that's what I'm saying here. So volume of interest, bounding box. That's a flat bounding box. So it has, since we did an annotation in a 2D section, it is a 3D bounding box, which is planar in 2D, yeah? But it already has a volume, so it doesn't have a zero volume, since we know it comes from the BigBrain and it has an uncertainty attached. This has I think this has now by default a 20 micron thickness. So it is already a box, yeah? Although it refers to brain section, but this brain section also had a thickness, right? So that's a box. And now, first thing I want to do is I want to get some context from the BigBrain. So I want to extract a chunk from the BigBrain model at a lower resolution in 3D, which just shows me some context in the BigBrain, yeah? Not yet the individual cell data. The way I do this here is I request the BigBrain template from Siibra in this one line. This does not yet give me image data. So the whole Siibra framework is implemented using a lazy loading architecture. So typically it will only load data when you really want to have it. Otherwise the whole system would be extremely slow with all the data that is included. So at this stage, I have just a semantic object representing the BigBrain template. It basically just knows where the BigBrain is stored, what it is, what is the resolution, things like this, not more at this stage. If I want image data, I have to fetch explicitly from this template. And what I'm doing here is I call the fetch method with an optional volume of interest. And I do not use my bounding box for that, since I wanted to extract some context. Instead, I take the center of my little bounding box and get the enclosing cube with 10 millimeter side length. So that's bigger than my annotation, right? That's more. And since this would already be in BigBrain, 10 millimeters is already a little bit something, I'm here requesting a lower resolution of, what is it, 320 micron. So if I run this, it might take, well, it was quite fast. So now I have actually, let me just look at this again. This cube should now be that's a nifty image now. That's a small nifty image with this cube, with a properly defined affine matrix pointing into the BigBrain space, with the proper resolution attached. So I could simply use this with all sorts of tools. I could even use that with the NeuroMap library, with NiLearn, whatever you want, since it's just a nifty image object at this stage. And that's not big. That's not as big as BigBrain. So now let's move on. And I could, of course, just look at it. So if you want, we could, with this, I could just, from NiLearn, there's the plotting library NiLearn, I could just say plot image. I think that's the thing, cube. And I don't want to have a background image on that. And here you see, right? That's the cube I fetched. I think I could even say view image. Is this, yeah. That's not Siibra, that's NiLearn. Great library. And that's interactive, so I can even scroll through it. Let me just get a gray color map that's closer to the original one. Yeah, so and here, you know, I can here move around. It's not a big resolution, but I see the surroundings. Now back to our actual patch. In the viewer, we saw that there's a set of 1 micron sections defined in BigBrain space available. We found one that intersects our region, and then we fetched, we zoomed into the whole resolution, in the full resolution. That's happening here. So Siibra has a module that's called features. I say siibra.features.get, and when getting features, I always have to do 2 things. I have to define where. That is typically a location. That could be a brain region. That could be a location object. In our case, we just use a space here. I say where, anywhere in the BigBrain. And then what? What I'm saying here is I'm interested in cellular features, specifically in cell body stained sections. What this will give me is a list of all the sections it found in the big brain space. And now I want to find the ones that intersect with the location that I have defined. And I can actually do this here. I go through all the sections, and I see if the location with which this section is anchored actually intersects my volume of interest, right? And I know that there should be at least one, so I check that here. And what I'm then doing is I take this, this section and I do the same that I did with the BigBrain template. I fetch image data. So I call fetch. This would fetch the whole 1 micron section, which is too much. And since I want the full resolution, I also cannot reduce the resolution. But what I do now is I fetch again with my volume of interest. Similar, very same pattern where I did it with the BigBrain template. I now do this with this 1 micron section. It is defined in the same space, although it is a section, so I can use the same kind of objects to define the volume of interest. So I use here the full resolution. -1 is a short form for use the highest available resolution, which would here be here 1 micron. -1 just says take the best resolution you can find. I provide the volume of interest, and what I get is again a nifty image, which hopefully shows me this 1 micron, this patch of the 1 micron sections sitting in the annotation that I provided above. So I'm doing that here now. Actually, in the next version of Siibra Python, which is already there, just not used in these notebooks yet, you wouldn't need to do the intersection yourself. In the newest version, you could right away run this features.get. Instead of putting the BigBrain space here, you could simply put your bounding box there, and it would already do the intersection for you. So in the next released version, that you wouldn't need these extra steps here. This might take a bit because now we have something at a high resolution. What I'm doing now here next is I plot both of them. And here you can see how the system safely uses the spaces. So I see here this 10 millimeter cube at lower resolution, and I see this 1 micron patch at 1 micrometer resolution, and I can just use the Nylearn library to plot them on top of each other, since these nifty image objects that it returned did appropriately set the spatial metadata. Yeah? So this fits together. I could even download the whole BigBrain and plot it together, and it would sit at the right position, right? I would just not see it, since it would then be so small. Of course, here I do not see the nice resolution of the patch, just because of the size. So I could also just use the array of the patch and just plot it as a normal image, but that's still the same data object. And you can see here, we have here a patch from a 1 micron section that was extracted according to my own specifications, which shows these giant beta cells that we've just seen. Not exact the same ones, because we didn't use the exact same annotation. Yeah. That's this one short notebook. Yeah. So this just basically, if you think back in the slides I've shown you, this mostly did the path here on the bottom, right? This part on the right. We could actually do the whole path on the bottom here by, instead of using an annotation, I could get a brain region from Siibra, then ask the brain region to give me its bounding box in a space, and then I could use this bounding box in the exact same fashion. So that would be just 2 more lines. I didn't do that for simplicity, but actually, so I can show you. So what we could do, for example, is Siibra get region, and then we could say we are interested in a region defined in the Jülich brain system, and we are probably interested in V1, left hemisphere. Oops, sorry for that. Left hemisphere, I'm running this now. This is lazy loading again. So here we have a region object. I didn't store it in a variable. That's the region V1 left. Let's do it that way. So you see now V1 left. That's the semantic object. It represents this region from that Jülich brain parcellation. So V1 left dot parcellation should show me which, that's from Jülich brain, and it used the newest version 3. So I see that here. I could then say fetch regional map. Then it will usually tell me you need to specify a space, right? So this map is defined in different spaces. So where do you want to have it? I could do it in the MNI 152 space, and then I should actually get a volume object. So it's now actually, since we are running in the cloud, as I said, lazy loading, it's now actually fetching first all the definitions of the maps that we have. Here we go. Yeah, and you see I get a nifty image. I did fetch as always, and when I do fetch, I get real data. So this is a nifty image of that particular map. Plotting, which I can again plot. Sebastian, I will speed up in a minute. And you see here's the map of V1 in the left hemisphere. Now I can, in a similar fashion, I can tell the region, get bounding box, and of course I also have to specify a space. And you see I get a bounding box object. And this bounding box I could use in the same way that I used the bounding box of my manual annotation, could do the same ways. The interesting thing is this bounding box here, you see, let me just do that. So B box of V1L. If I plot this, if I just print this, what did I do wrong here? Oh, Books. Books. That's a nice name. Books. My next software will be called Books. So you see here, this is a bounding box in MNI space, right? If I would now query, I don't dare to write this down now, but if I would use Siibra.features.get and use this bounding box, what Siibra would actually realize is, oh, I have these 1 micron sections, but they are defined in a BigBrain space. And this bounding box is an MNI space. So it would automatically warp the bounding box from the MNI space to the BigBrain and use it there, right? So in the background, what I can always do is I can say bounding box V1L warp to BigBrain. And of course I have to print this. And you see now I get a bounding box. It has different coordinates here, different coordinates, and they are attached to the BigBrain space. Yeah? And Siibra would use this in the background. We could do the exact same thing, searching for 1 micron sections in that intersect V1. Yeah. So just to round up this a little bit, you typically, in Siibra very often you start with some concept of location. That could be an anatomical structure, it could be coordinates, different things, and then you start placing them in an appropriate space and doing things with it, very often querying data. Yeah, so just that you see that this is not, you don't have to remember all these things. There is a documentation on Read the Docs as you would expect it. And the most important part of the documentation is there are working code examples, documented code examples that will be automatically generated with each version we ship, where you usually find these things. So for example, there is a category maps and templates here. And there is access BigBrain high resolution image data. And when you click there, you find a working code example that does more or less the things I'm showing you here now. Yeah, with images, and you can then download this piece of code. So you see here, even we here even downloaded the chunk with the layer segmentation for the same thing. And in the bottom, well, it's actually even shows us a 1 micron patch. I wasn't aware. And you can download then here the corresponding code. And there are many other things. For example, there is here a whole category about multimodal data features. You remember that Sebastian showed you in the viewer how when you select a brain region, you find connectivity data for that brain region, or you find data from other histological experiments. You can also find this stuff here. For example, there is also here a code snippet that shows you how you can access receptor density information as for a region. And you see here, let me just make that a bit larger. You see here that this works exactly the same way. So I call zebra.features.get. That's what we just did as well for the 1 micron sections. And then again, I ask where, and here I put a region. Yeah. So I'm interested to search for receptor data in V1 according to the Jülich brain. Here I also have a version specified. And then I give a different modality here. So the what is a different one. It's molecular features, receptor density fingerprints. And then this will give me the underlying data that we also saw in the viewer for these receptor measurements, which will here in Siibra typically be represented as a pandas data frame, which is very easy to reuse with other tools. Yeah. So much of what Siibra gives you are nifty image objects, pandas data frames. There are not, that's mostly it actually. So most of the stuff is in the end in these formats. And the idea is always that you get this data element late according to this lazy mechanism. So we also tend to provide access to the nifty images very late, because once you deal with the nifty image, you lose the connection to the reference space. So when you search for a volumetric data in Siibra, what you usually get is a Siibra volume object, which has all the links to the reference spaces. Only when you say fetch, then you will get a nifty image and you lose that connection. Yeah? So we usually promote to do that very late, so that you keep the data in your process, in your workflow, as long as possible attached to the metadata and all that. Of course, at some point you really want to have the voxel information, and then there's no way around. All right, with that, let's maybe go back into the viewer, yeah, and look at a few more features. Yes. Yes. So, yes, I go to the slides. So we have shown you in this notebook how we actually get down to a 1 micron patch, right? Of course, you're usually not super interested if you do a study in this image. For these types of images, what you would be interested in is actually how many cells are there. How many cells per layer do I see, you know? And is this representative for that brain region, for example? And that would mean, of course, that you start now using different tools and actually analyze this patch. But since this is important information about brain organization, and since our idea is to provide a lot of information about brain organization already in the atlas, we have basically used this principle to prepare data sets for you, which already have this regional information ready. And we have that here. Ist das F5? Yeah. Funktion F5. Ah. Okay. So what we have done is we have used the exact same principle and extracted cortical patches in a selection of cytoarchitectonic regions. For now, what we have published are 12 of those regions, but I get back to that. And we have extracted such patches, I think at least 10 per region from different sections. And we have annotated manually and very precisely with a 4i principle quality control the cortical layers with more precision than you have it in the BigBrain cortical layer atlas. And you see that here. And then we have used a recently published very precise cell segmentation algorithm to actually extract the shapes of the cells that we see there and the areas, and have computed the histograms of cells with their different sizes in the different cortical layers. So you see this is here a little bit of an animation of this data set. You see on the top the points where we have extracted patches in the big brain space, and on the bottom you see these extracted cell numbers in the different cortical layers. What we have used here is a recent deep learning algorithm. I have to show it because it has been developed in my group by Erik Upschulte, and it was very successful at NeurIPS in a benchmark last year. Actually, we made the second place there. So we have used this state-of-the-art, really precise deep learning algorithm to extract the cell bodies. And now we have worked, so these 10 brain regions are already published. You can access them in Siibra. In the interest of time, we don't go through all the code snippets, but here on Read the Docs, it should also be there. So you see here cortical cell body distributions. There's an example, and you see that it does exactly the same things we have seen. Siibra.features.get, we provide a brain region, and then there's a different modality also in the cellular category, cell density profiles. And they give you these precise densities of cells in the different cortical layers, but of course they give you also directly access to the exact segmented cells, the individual cells. And these data sets are also, as I said in the beginning, everything is a fair data set on EBRAINS. We hope fair enough at least. So if we go to EBRAINS, search, just should know the right keyword now. I think layer-wise is one of the words, no? Layer specific. You could also click through the modalities and all of that. You see here, here's one of them. Layer-specific distributions of segmented cells. You see here the data set with all the information. And if you fetch that data set in Siibra, you will of course find the DOI that brings you to this data set again. As I said, it's usually is always mirrored. If you find a data element in the atlas, it will be mirrored in a knowledge graph. We are currently we go back into the slides. We are currently working on update of these, as of now, 120 samples in 10 different regions. We have been working on much more. So the red dots that you see here is the currently released data, and the blue dots is what will be coming very soon. We have done this now for, is it 90 different brain regions?
78 are coming in 12 regions, so we'll have 90.
Yeah, so there will be samples of such cell densities in 90 different regions with a very good coverage of the whole brain.
We're dealing with the view stuff, so
through Siibra and the EBRAINS knowledge graph.
I have a question for you.
Okay, that's the cell densities. At this stage, do you have questions? I'm not so sure whether I'm going too fast or not.
But from a second computer.
It's hard to see your faces, whether you are struggling or sleeping or being attentive. Okay. Then I would say we go back into the viewer and have a look at the,
so I would need another
receptors again.
So,
yeah?
so it's
would you
not
want
planned,
to do that?
But it's just in case. Mona,
so, here we are back in the viewer. So we have some 20 minutes left, and I have some ideas what I can show you, but it would also be possible to adjust now. So if you have specific things that you're interested in,
how can we tell
I'm
if
totally happy
to
prioritize this. If not, we will definitely go into something that we prepared. Okay. So as we are here in the big brain, maybe one thing that I would like to show you before I go back. They are not only sections anchored, so we are also working on volumetric reconstructions at higher resolutions. For example, if we go here into the occipital part of the big brain.
Where should it be?
Oops, wo hängt der denn hier?
Okay, yes. There it is. Yes.
If we go into the occipital part and we look here at the features anchored,
Right.
you see that there's not only sections, there are also some volumetric data sets anchored. Here's one of them.
Okay.
So here's a 3D reconstruction of a tissue block from the big brain, which was scanned at much higher resolution, which is also published in EBRAINS. And you see here that's actually a stack of one micron scans reconstructed at high detail
Make these
in
boxes
3D.
as small as
so
possible.
you get here quite nice, with some missing sections in between, but a quite nice higher resolution representation of this block in the occipital part. And we are filling in more and more such 3D data. So this uses really a search by location, spatial search. There are also a few things. For example, in the hippocampus, we should find a few interesting things. I guess that's here, the hippocampus.
That's good.
I'm not an anatomist, so forgive me. Was hat er jetzt hier so reingezoomt? Wie kriegen wir das wieder weg? Well, so there should also be something. Es ist so reingezoomt hier irgendwie, das ganze Ding. Also der Viewer, nicht der Browser. Naja. Nee. I'm not so familiar with Windows, but it somehow enlarged the whole window now. Maybe switch forth and back. Does that help? I think I did a double click here. On Mac it does it as well, but then there I know how to get back.
I'll probably just connect the
Here I
second
don't know
computer
it.
if needed.
Well, so here, for example, you also see some information from polarized light imaging in the hippocampus. Also, volume of interest that has been anchored here in the hippocampus. So there are some things to find by browsing the 3D anatomy. Okay. Let me maybe go back to the now we are back here in the MNI space, one thing we also wanted to mention that a typical way of using the atlas would also to explore how features differ in different brain regions. We saw these receptor densities. I think in the exactly a few minutes ago we looked at this area 44, which is a language region, I think, and we showed you here how you can access this regional information. For example, the receptor densities. And if we look here at the GABA, not sure if it's GABA A or GABA B, maybe it's GABA B is the more relevant one. Let me do that. So I look here for GABA B. Here we go. So here we have a density profile representative for this brain region of GABA B. And we can see, is this really the one?
Is there a way to blur my background?
I selected GABA A again. It's too late. Then let me do that again. That's GABA B. Yeah, so here that's representative. And what we could see here is that, you know, that basically this plot shows us the measured density from autoradiography experiments going along the cortical depth. So we see here that there's a peak at the lower cortical depth at somewhat clearly above 3,000 femtomole per milligram. Actually here 3,406 if we look exactly. We could of course download this as a CSV data item here from the browser. And it might be interesting to compare this to other brain regions. So we also, before we went into the big brain, we also looked at a motor region. Anyone remembers the classification of this? The region name? I think it was 4P, right? 4P left. So we could also do this.
For now.
And have a look here at GABA B.
Oh, sorry. Okay, I closed it. Okay.
And one thing that we didn't really show, as I said, it is always a data set in the background. So here you see again a DOI, right? Just as an example, we're now looking at this receptor density data set that is linked to the region 4P. Here's again a DOI. So if I click on that, takes us to the data set in EBRAINS. We see the authors and all the, wow, this is going slow. Here we are. So we find here all the information. There's a data descriptor and data, and should be everything there. But we can then just here have a look at it, and what we see here is that the concentration level is lower here in the motor system than in the language system. Yeah, so this is something you could here easily see just by exploring that. And you might be interested to see whether such a difference can also be observed in different data modalities. And one interesting thing is, of course, transcriptomics. So this online viewer also has a plugin implemented that can interface to the transcriptomics data in the Allen Adult Human Brain Atlas. And we could use this to do a similar study here. Maybe at this stage, Sebastian, would you like to explain something about this JUGEX tool? So this is a plugin. The viewer is extensible with plugins. And this plugin uses an existing analysis tool for analyzing gene information.
Maybe differential gene expression. So, of course, it's a bit constructed because we want to show something which works. So, but at the end, what we want to show is that we can use, on the one hand, receptor densities as a modality, and we can directly compare it using all the modalities which are included in the online viewer here. In the tool, there's a plugin. It's called UJEX. It's only an example. So you can, at the end, write own plugins and go with those plugins to Timo, and he will try to help you to introduce or to bring those plugins into the Atlas viewer or into the Siibra family. And Siibra UJEX is one of the plugins which is already there. And you can select areas here, like the same way we have done it before. So you can select them by the name if you know it, and we know it. And for example, the area 44. So those are the two areas Timo has showed you with the different receptor densities. Then we select genes of interest. So those are the genes which were analyzed by the Allen Brain Institute. So those genetic data, they are coming from the microarray study, which were published, I think it's about 5 to 10 years ago, I'm not sure, sorry. So they have analyzed about 3,600 tissue samples scattered all over the brain, and they have completely analyzed the genetic information from those patches, and they also brought their data sets into the MNI reference space, and therefore it's very easy for us to directly grab their information, their tissue samples, which are located in a specific volume of interest here in the different areas. And I think here, this is a good example for really good metadata, because the Allen Brain data sets, so the tissue samples in the microarray data set, this was really described very well. So it was quite easy to take the data, to understand the structure of the data, and therefore it's really easy to work with their data and create your own benefit from the data from somebody else and analyze it in your workflows and in the way you would like to do it. So now we are here looking for two genes which encode for the GABA-B receptor. As I said, it's a bit constructed, but okay. Yeah, it's a normal plugin. You can do it here. It's a normal permutation test. It works exactly the same like the null test we have seen in the workshop before. So it's also a permutation test. Here, also with only 100 permutation, this does not really make sense. So, but we have placed a very low permutation value here because otherwise it simply would take too long to calculate it. And you define a threshold. This is for the volume of interest. So as I said, the maps are probabilistic maps. They have values from 0 to 1, depending on the probability of being the specific area. Here we select simply threshold. You can directly run it here in the browser and visualize it, but you can also here again switch to the Python world. So therefore, I think it's a really good example because it shows the direct connection of both sources. So you have selected the region of interest in the volume in the viewer, and by clicking Notebook, it will bring you to a Python notebook, which can directly be run here in the browser, and all the selections you have made in the browser are included here. So here we have the two areas we have selected, we have the two genes, we have the thresholds, we have the 100 permutations, which here would not make sense. But this is a really good starting point for everybody who is not familiar with Python. But yeah, it's easy to access the data sets through the browser. And this helps you to get a starting point here. And all the things Timo have shown, plotting, whatever you would like to do, transferring your data to different spaces, this can all be done here in this notebook as well, or you can download it and run it on your local version. It really depends on your need. You can try to run it normally. Didn't we have it open somewhere? Do you remember it? Oh, yeah, here it is.
But that's also
Yeah, this was here. Okay, yeah, we've loaded it before. So, but if you press start from the page we have just seen, you can click through this notebook here, and it will install Siibra here in this virtual environment, load all the necessary dependencies, and then it will simply start the code you have just clicked in the browser and give you the results of the permutation tests. And here we can see that for the one gene, GABA-R2, it's more or less significant, but of course with 100 permutations, so that's definitely not something you would like to publish. But just gives you an idea of the things you can do with this format. And of course, you can provide a complete list of genes. You can merge different areas and do the analysis for different areas you would like to, for different pastillation schemes. You can plot the positions of all tissue samples and compare it to the probabilistic map in the background. And this is something you can really use for scientific publication. So you have a working script which can be put into a manuscript, and it's really reproducible. And all metadata and all information which are available can directly be accessed through this notebook here.
Yeah, I should maybe. We had actually planned in a bit of time for questions, so we are more or less through with the content. I was really hoping also to discuss a bit more questions with you. One thing I wanted to say that what you just seen is something that we would like to promote more with this system, that you can use the visual tool to basically see what's possible, to find an easy entry point how you work with the system, but then have a very easy transition into the reproducible world. And I find this quite a challenge, to be honest. But we have here, for the first time, basically implemented this idea that everything you do interactively has a corresponding programmatic, a sequence of commands in a programmatic way. And we want to push more in that direction. So the idea is that there are even more places in the online viewer where you do something, and you could actually get the Python code snippet that does the same thing, but that you could put this in a script or put with your publication, because there's actually a one-to-one correspondence with everything you do in the viewer. It's only not yet the case that you can click and you get the Python code for this one-to-one correspondence. I'm actually quite passionate about getting that implemented. We did it now for this Jülich plugin, but that is really an idea we have that you can access this quite easily for many things you do. It's quite a challenge to build that into the workflows, but that's really the idea we have. And I would also like to make a second and last remark. As of today, you might sometimes, if you start using that now, please feel free to do this. So we are here still pre-version 1. So sometimes we still have breaking changes. And just this week, we have been updating the Python code. So it might happen that when you download a notebook today, it's defined for the previous version and not for the one that's out right now. This is something that we are hoping towards the end of the year to arrive at version 1, where things are stable and where we are not changing this anymore, but usually you get messages about these things when you use the tools.
And I think the connection between the two worlds, between the browser world and the Python world, goes also into the direction we have heard this morning in the lecture, that simply pointing to a manual normally is not enough. So if I have a question about some specific stuff which is included already in Python, Timo already says it's very easy. You simply have to write, and he starts coding something, and after the third line, I'm out because it's too fast for me. I cannot code that fast. But if you get an example of a running notebook which presents your code which is already working, this is a very helpful starting point from our point of view, because you can directly start, and adjusting smaller points in a running script is a lot easier than starting from scratch and installing everything and really learning it from the manual. That's why we also think it's a very important point to bring those two worlds together and make it easy for the user.
Right. We're at the end of the tutorial with this sentence. So we have a few minutes left. So if you have a question, just go ahead.
We are the first at the coffee break?
Yeah, otherwise let's close this.
Oh, there's a question, Timo. No, there's a question.
Oh, yes. Great.
Hi. So my question is it possible to use the Siibra Python framework to, you did some, you create a bounding box from the Jülich brains, the Sebastian atlas, and then you warp it to the BigBrain. So then is it possible to export, to fetch that bounding box with the Gifty file defined by that bounding box, like a volumetric data?
You mean a Gifty file of the surfaces or volumetric file?
Any surface or volume
Yeah.
data.
So the question is it possible once you have that BigBrain thing to store a Gifty file with that information, right? Did I get it?
Yeah, so the last step that you did was warping the
Yes.
bounding box from the Jülich atlas
Yes.
to the BigBrain.
Yes.
So my question is, with this warp within the BigBrain, can we fetch this as a surface or as volumetric data?
Yes, this is possible. So you could simply fetch with it right away. You wouldn't even have to warp it. You could. We had this line with the BigBrain template. Let me just show that. I think it's still. we opened so many things here. Yeah, here's the notebook. So you saw here we fetched, for example, this 3D. No, that's not the one. Here we fetched from the BigBrain, right? You could, here, for the volume of interest, you could directly put in the bounding box of the brain region in an I space. It would automatically warp to BigBrain space and fetch what's in there. Then you get a nifty object, and this is an object from Neuroscience, which already has a 2 file name, so you could store this as a nifty file into disk right away. Right? So that would work. You also mentioned Gifty. I should admit that Siibra implements a whole class hierarchy for volumes. And until now we focused a lot on voxel volumes. So it also has support for surfaces. You can work with surfaces and meshes, but it's not as strong there as a volume. So you could even also get a surface mesh and store it, but as of now you cannot cut surface meshes and extract parts of surface meshes. That is something that will certainly come in the next year, but we have not spent so much time on making things work for surface meshes. Yeah? So you will miss a few features. It will typically let you know that's not implemented, but things work better for voxel data at the moment. Yeah. But the idea and the workflows and the names of the functions are the same. So if you fetch a surface representation, I could say Jülich get map, and instead of MNI 152, I could say fs average, and I would actually get a labeled surface. That works. Yeah, and this is also a volume object, just like the BigBrain template is a volume object. But when you fetch from it, you don't get nifty. You get a dictionary of vertices and faces and so on. And with this one, there's not so much support to manipulate it. Just to be very honest. Yeah? But it's coming. And if you're interested in these things, just drop us a note. We're happy to help. More questions? Then let's run for the coffee. And thanks a lot for your interest and your patience.